in territory, continuing in my movement organizing series, Thursdays, roughly between 10 and 12 Mountain Time. Here we are, wherever you're seeing this. Today won't be interactive, but if you do leave comments, I'll get around to them wherever you're seeing this. Moshe Feldenkrais is, for me anyway, not that quotable of a person. It seems that, for me, most of the time people take a quote from Moshe Feldenkrais. It's the context. Without the context, the meaning suffers. Sometimes it means something almost paradoxical in outside of its context. And movement is life is one of those quotes that there's a couple reasons for it showing up in such quotable forms. One reason is that Moshe put a quote of it, a quote, something to that effect. Movement is life. Without movement, life is unthinkable. I think it was that version of the quote that's on the Learn to Learn pamphlet, I think it was called, or I can probably dig it up. I know it's online somewhere, but a kind of Intro to Awareness Through Movement pamphlet that Moshe Feldenkrais circulated with his first recordings, I think. But so if Moshe takes a quote of himself and puts it, or takes a phrase and puts it in isolated context, then it's supposed to hang there and be interpreted on its own, like an aphorism, the kind of stuff other writers or philosophers have contributed over the years. Some authors are particularly famous for their pithy aphorisms, and then some authors really aren't or shouldn't be. They shouldn't be necessarily digested down into aphorisms. Einstein is one of those people that gave great kind of one-liner quotes, so great that there's almost a genre of bullshit Albert Einstein quotes that are they sound kind of in a modern physics vibe, a kind of relative postmodern vibe, maybe something consistent with what a pop culture understanding of relativity, but are not actually Einstein quotes at all. I'll hide my image for a moment. So yeah, we're working with the movement as life theme today, and it's going to be maybe mixing a little bit from the other themes of the week, which, if you've been watching, weren't actually streamed this week, namely the movement structure theme, where I'm reviewing anatomical ideas and the relationship between anatomy and physiology. And then in the Wednesday movement learning theme, reviewing even drier materials related to physics and math and statistics. We're sort of drawing on all that today because a lot of my background is specifically in this area, the science of movement, although at different scales, at different spatiotemporal scales than human movement. What we mean by voluntary movement, the kind of stuff you can see a person doing in front of you. What Moshe means by movement is life is interesting. Again, there's more than one flavor of this quotation, but one of the flavors of the quotation follows with this statement that life is a process. And so Moshe is trying to actually say something about life itself, life in general. And in that sense, what's true of what's that that quote, what's true of the E. coli is true of the elephant. That quote from Jacques Monod, the Nobel laureate physiologist, I believe, who was discovering a lot about the basics of life by studying microbes. And the idea in studying microbes isn't that they're equivalent to multicellular, macroscopic, familiar organisms like elephants. Not at all. Where's the feature to loop this? There it is. The point is not that what's true of an elephant is true of an E. coli. The point, or a bacteria, I should say bacteria. The point is that what's true of a bacteria is true of an elephant. So there's an important directionality to the logic of what Monod is saying there. So it's a Jacques. I think I'll go back to white. I think it's Jacques Monod. What's true? ... E. coli ... elephant. You can find the quote yourself, but the point is that what's true of the bacteria is true of the elephant. What's true of the bacteria is what's true of the elephant. Not the other way around. So bacteria, for example, has things like cell division, homeostasis, and all these processes are present in the elephant. They're still fully reflected there. You can say migration, movement. We can put movement in there. What else? Signaling. We can put that in there. Now, bacterial signaling is going to be a little different, but there's still even bacteria detecting that they have groups of each other so that they can cooperate and differentiate their functions. So, yep, signaling, all these processes are there. And then in elephant, we can even, well, we have cell division, but we can also have mating in there. Let's have mating in there so we get the richness. And feeding, we didn't write feeding specifically. Feeding, okay. So all these things are true of bacteria, and the way that bacteria do them are even the same way that elephant do it. What? That's true. Everything that the bacteria does is done in the elephant the same way. Just digest it for yourself. That's the nature of things. And then what's not true is that what's true of the elephant is true of the bacteria. So how the elephant feeds is not the same way the bacteria. The elephant feeds with a trunk and a digestive system and a whole bunch of complicated apparatus that a bacteria simply doesn't have. So the elephant has all kinds of manners of feeding that are not analogous to the ways that the bacteria feeds. The elephant has all kinds of ways of behaving. So what's true of the elephant is that it's got an emotional life. I'm not going to say who's experiencing what or who's conscious, but the bacteria, the elephant's got yeah. I won't even actually use that functional of a language, but I'll say the elephant's got a neuroendocrine system. So it's got a nervous system and an endocrine system all together. And so it's producing neuroendocrine system events. These events are not analogous to what happens in the bacteria. They cannot be, right? So again, I'm not talking about is the bacteria aware, or does it have a mind, or is it conscious? I'm not actually going down that rabbit hole. I'm not using that language. I'm just saying that it doesn't have the parts to do the business of all the business of being an elephant. Elephant business is very serious business and involves waving your trunk around and sucking up water and spitting it out and snapping around and being an elephant. I don't know. I'm not an elephant exactly. I'm not trying to be disparaging of all the elephant, ways. I do not know the elephant ways. However, I know that they are rich and complicated and necessarily more intricate than the bacteria ways. Should that be such a controversial statement? It turns out that for some people it is. And if you feel any dissonance with what I'm saying, then I really suggest that you follow it internally. You follow in yourself where the resistance is coming from so that we can talk about it more. So there's a one-wayness to these things. Oops, didn't mean to look at that one yet. What am I doing? Oh, went on to the other video. I see. It's cycling between Amoeba and the flycasting video. So, excuse me. I think I should take this phone call actually. Ah, they hung up. So I took this video of a paramecium hunting, sorry, an amoeba hunting paramecia. When I was teaching introductory biology in University of Baltimore. And so it's a little, the contrast maybe isn't great. I need to get this second video out of the queue here. 1 second. How do I do that? Geez, I need to at least turn the sound off this thing. What kind of half-ass live streaming is this? I'm fired, right? So is this the playlist? Yeah. All right. So that's a paramecium that's getting actually engulfed by this amoeba and eventually endocytosed and digested. I don't think the actual endocytotic event is on camera exactly. Yeah. Oh, maybe it is. That's still the paramecium there in the vacuole. I don't know at which point it begins becoming actually digested. How do you feel watching this video of the amoeba hunting the paramecium? Do you wonder there's the paramecium and it's figuring out how to engulf it and then it does it? Is there something about the narrative that I'm presenting? There's a happy free paramecium. There's something about the narrative I'm presenting that's challenging for you. Let's let's locate these points. Not from me. I don't need to actually convince you of anything. I'm not motivated to teach people that there are such things as amoebas and paramecia. But this is one of these everyday miracles that you get to observe if you are privileged to have a microscope and to have samples of things that are identified for you. I was actually in a student laboratory and had like a prepared vial of this stuff that we bought from another company that they sit there and grow these things and prepare these samples to show so people can look at it under the microscope and go wow. But all I had to do is manage to pipette the right bit out of the thing into the onto the microscope slide stage, and the university had already given me a microscope with the camera attached, and it was easy from that point. The hardest part was finding an amoeba. Even though they gave you the jar full of it, I still had to pipette. Like they kind of hang out on the bottom of the jar, so you had to learn how to get lucky, how to hunt them, I guess. Um, but you can't see them. This is under, I don't remember the magnification, but at least 100 times magnification. Um, you can barely see the amoeba with the naked eye if you use a bit of imagination. They're I think comparable to human ova if they're in a round situation in size, um, but very small things. And who can who can dispute that there's something alive going on here, that there are life processes going on? The paramecium, owing to its mode of locomotion, doesn't necessarily appear to be alive at the scale. It might just be tumbling randomly. You can't see that it's rowing itself, that it's got hairs that are moving itself in a specific direction. Um, but the amoeba is it's pretty compelling. It's hard to look at this thing moving around and to come to any other conclusion than it's alive, because it's not only changing its shape and doing things, but it's doing it in a kind of intentional way. You see at the start of the video here, when it gets a hint of this paramecium, it then decides to flank it. It seems to know to engulf it, how to move around it. And it's three dimensional. It's building an actual bubble around it that eventually seals around itself and forms an enclosed space inside the amoeba. And then the amoeba just needs to remove the non-paramecium bits from that space and to introduce the ingredients the ingredients required to digest the paramecium itself. I'm not an expert in this. This is just basic cell biology. This is just more or less how it must go. Um, and I'm not even an expert on movement at the scale, although I have a lot of life history, a lot of experience working with the detail of the machinery of movement, but in the context of animals, not protists, which is what we're looking at. These aren't considered animals. These are single cell organisms that behave in a way that is so animal like that they immediately resisted classification as bacteria or plant life. And they've continued to resist being, um, well, now after the tree of life has been more subtly, compellingly laid out with molecular genetics, um, it's understood how closely these things are related to us, even even though they're not even animals. With no nervous systems, they nonetheless are moving using similar tricks to the cells in our body, and some of their tricks of movements are recruited by tissues in ourselves. So, for example, the cilia are swimming using, sorry, the paramecia are swimming around using oars that are uh, that are cilia, that are the same structures that are lining our respiratory tracts and that allow mucus to move up from our lungs so we can hack stuff up regularly. Um, Cilia are probably used in lots of other locations that I can't remember. Um, and for the paramecium, there's cells in the human that actually move in a very analogous way to an amoeba, like white blood cells will do the same thing that the amoeba is doing. Literally, they will hunt other non-human cells using the exact same movement pattern as amoebas. So, what's true of the amoeba is true of the human. Um, I think I found from some online biology website um, this image of a paramecium. Oh, I should leave the. It's Byju's is the name of the app. It's a learning app. Thanks, Baiju. This has to be fair use if I leave your logo on, right? So there's your. Oops, didn't mean to do that. Here's your paramecium. So yeah, it's covered in cilia. There we are. They're labeled cilia there, these little hairs on the outside. And they row. They're actually like oars which pulse in a directional way. But it appears to have a mouth end, an oral groove. It's actually got a shape to it. It's got um, a nucleus, which is specialized in different parts of the cell. It's got things like a kidney. So yeah, it's got all kinds of things that recapitulate the processes in animals. And no cell in a human looks exactly like this, but this is a one-celled organism. But what's true of this one-celled organism is also true of the cells of the human being, like as in the notes quote there. Um, so okay, so oh, I wasn't even showing you. Sorry. Oh, why won't this? Technical problems. There you go. There's your cartoon of the paramecium. So that's the thing that the amoeba is eating. And there's the amoeba. So not the scale. The amoeba is much bigger than the paramecium, as you saw. But same kind of business. Specialized structures within the cell because it's a one-celled organism. It's doing all the things that a whole organism does. But just a cell nonetheless with different parts. This is the thing we see flowing around here. An amoeba doing its amoeboid motion, actually making some parts of itself rigid and solid like a sol state. No, a gel state, and then some of its parts fluid and flowing, the sol state. And just by alternating how liquidy or jelly some parts of it are compared to other parts, it flows into different areas. Well, it does all kinds of other things. I mean, in altering that liquidness, that gelness, that's quite a feat. And it's mediated with the same kind of machinery as in muscle cells. When I say machinery, that's a shorthand for the structural proteins that actually do things. What we're looking at here is the microscopic scale, but it's still enormous compared to the size of molecules and the structure of the parts of these cells that actually interact with each other to make the specific functions. And so, although this scale isn't familiar to people without access to a microscope, um, the the quality of the movement itself doesn't seem too far away from what we're familiar with. Things are kind of floating and drifting. There's a quality to the movement that is analogous to things that we're familiar with. The bobbing and the bobbing of objects that are floating in water, the way that fluids flow, and it's really just the specific quality of amoeboid movement, how it intentionally generates arms and wraps it around things, that makes the movement here not human in quality. There's no skeleton. It's not organized in any way that's easy to anthropomorphize. Oh, I'm glad I mentioned that word because this business of thinking about intentions and thinking about the life of an elephant as being relevant to the life of an amoeba, that's anthropomorphizing. When you go in the other direction, when you go in the other direction, oh, when you go from the elephant direction to the bacteria, that's an anthropomorphism because you're or an elephantomorphism. You're saying that the life of the elephant is analogous to the life of a bacteria, and it's just not the case. There's no reason to think that. So whatever we mean by experience, whatever the experience is of this amoeba, there's no reason to compare it directly to the experience of a person. And when I say hunting, it's because it's an apt functional description of the behavior. But it's not sitting there with an orange hat in a duck blind drinking beer. It's not doing those things. There's the only thing that's analogous about it is the actual specific functional behavior, but the intention we don't know about. How it intends, what it means for it to intend to engulf this paramecium, we don't know. And of course, from the perspective of the paramecium, maybe we're feeling empathetic. We wish that the engulfed paramecium over here was living free and happy like the one over here, and I keep not flipping back to the video. The engulfed paramecium now over here is not free and happy, and it's easy to put these this kind of language on the situation, but we just don't know, and there's no reason to do so. it's very hard to explain what the experience of a paramecium would be at any point. And we have a hard enough time empathizing with each other and connecting with the natural world by imagining that it's got a Disney personality to everything that is an echo of a reflection of the human mind. It's not it's not a helpful or sophisticated worldview in my perspective, and I don't think that it's at all what the animist cultures are doing. They're not. I don't intend to speak for them, but I'm also not trying to criticize them. I'm criticizing um, the Disneyfication of everything. So, how this movement actually occurs is another question. Where does the movement come from? Why don't you meditate on it for a moment, like a koan? Where does the movement come from? You can dwell on it how you want. Where does the movement come from? You can look at this video and see all the things moving around. Why is it happening? What is the source of the movement? These are microorganisms that are sitting in basically water on a microscope stage. They're not being driven. There's not like a large prevailing current. There might be a small circulation within this thing that's helping the paramecium have a net movement this way. But again, it can row in a direction, and there's no current making this amoeba flow how it's flowing. It's somehow making decisions for itself, and it's moving in particular ways. How does it create its movement? Well, I kind of blended the scientific and philosophical framings of the questions because I'm not interested in being formal or impressing people with formality, but rather connecting with what your actual experience of this is. And if you're a Feldenkrais practitioner, go ahead and do bell hand for a moment. Let your fingers come together as you inhale. Just just do the right hand so you can really focus your dominant hand. Just focus on the fingertips sliding over some surface. In response to this other process, the inhalation, as you exhale, the things move the other way. And that's voluntary movement you're making. Sure, it's in response to your breathing, which is not exactly voluntary. But what you're doing with your hand is a fully voluntary movement. Where does that movement come from? How does the movement happen? Is closer to the scientific framing. Why why does movement happen? Is a less scientific framing. Why does the movement happen? But there's this simple answer that you might think I offer as the ready-made answer, the con, contractile proteins. like actin and myosin. Um, other sources, and what happens next in the little shared today, it isn't the answer to this question. I'm offering it as a koan, or I can give you the answer right now because I'm not trying to lord it over you, but I really think the literal answer is that the motion comes from thermal thermal energy, from um, from heat energy, from disordered energy is where the movement comes from. And so that's the actual bottom of the cascade. And the trick that's being used to intentionally move is the same kind of trick that cells are using over and over again, very akin to fishing, trying to gather up randomness into useful little chunks and then use the randomness to do things. But take a pause from that question and look at a video from the late and great Susan Lindquist, an early inspiration for me, made huge contributions in protein science around the area of prion biology. And so you can find Susan's lectures still on YouTube, at least some of them. And so this one's from November of 2013. I need to make sure the sound's coming through. 1 second. Let's try that.
A variety of different biological problems, and they all have one thing in common. They have to do with the problem of protein folding. So what's the problem of protein folding? Well, proteins do just about everything in our cells and for us in our bodies. When you think about seeing an apple on a tree, the light that you perceive that apple by is being harnessed by protein receptors in our eyes. And it's a series of actions of various proteins that transmit that signal from our eye into other parts of our brain that can wind up interpreting that image. When we go to pick that apple, our arm is powered by muscle. Muscle is protein. The engine for creating the energy, the mitochondria inside of our cells, are made up of thousands of different proteins that are involved in providing that energy from that movement. When we bite the apple, when we taste it, the taste is being perceived by receptors on our tongue and in our nose that are made up of proteins. And when we digest the apple, the enzymes that are involved in digesting it are proteins. So proteins are us, I guess, is the way to put it. And proteins, however, are encoded by DNA. DNA is the code of life, the double helix. And DNA is linear. It's a long, very complicated code, but it's sort of like not really, in some ways, all that interesting in and of itself. It is the information for life, but rather like a cassette tape. It can play different tunes depending upon what part of the DNA is being accessed. But the tape itself is linear. Now, the way this DNA plays different tunes, basically, is that it's decoded into long linear strings of amino acids based upon the sequence of nucleotides in the DNA. So here are two very different proteins that are being made by these different parts of the DNA. And in order for those proteins to function, they have to fold up into these incredibly intricate, complicated shapes. This is hemoglobin here, the protein that's involved in carrying oxygen around in our blood. And this protein over here is green fluorescent protein.
So I just wanted to pause there and just because a lot's been compressed into that introduction, but here are two different proteins, and so these are single molecules of proteins, and each molecule is associated with a different role or function in a different biological context. And so the one on the left is a human protein that is used for transporting oxygen through our bloodstream. And it's related to a lot of other similar proteins that are in muscle and things like that. And so the movement that hemoglobin's involved with involves movement through the circulatory system and movement of oxygen molecules and also things like bicarbonate onto and off of the hemoglobin surface itself, onto its heme groups. And then hemoglobin itself changes its shape. It actually moves and alters its own shape in response to these different events. So there's movement of all kinds and all qualities that hemoglobin itself is part of. One part is a movement which is related to hemostasis, or like the movement of blood through the circulatory system. And so it's the kind of thing we're familiar with. You can see the blood dripping out of an open wound. You can see it squirting into your sampling vial and get a little bit nauseous, maybe as if you're me. You can you can have an idea and a sense of the movement of blood through the circulatory system, at least part of it. I mean, discovering the circulatory system itself, it required a microscope. There's a reason why the arterial circulation and the venous circulation weren't understood to be connected to each other through capillaries until the invention of the microscope. Because you can't see the capillaries, you can't see how the arterial blood supply transitions circuits into the venous blood supply. Again, some aspects of the actual movement changing the quality as the scale changes and it becoming inaccessible to human intuition. At least the kind of intuition patterned on everyday macroscopic events. Then this other protein, I think, is from a jellyfish, and it's a fluorescent protein that's useful for things, but not really used in our physiology. And then so the other thing is that these are single molecules again. They look like very complicated structures because they kind of are. And we're only looking at a small fraction of all the atoms in these things. They're being cartooned into simple ribbons that only trace a small set of all the atoms in the protein. And so you can see one end of the molecule here is going up, and this is all folded together. And then the other end of the protein, I can't quite make out where it is. It's typically pointing in the opposite direction somewhere for interesting reasons. But nonetheless, okay, so then the hemoglobin's made of two different protein molecules that come together in a group of four. So this is four protein chains grouped together, the red and the blue ones. This is, I believe, one continuous protein chain that has a little bit of modification to it as it folds. Nonetheless, it's like origami. It's like you took a piece of paper and you just need to make folds and you get these shapes. And so if you pulled on one end and the other end, you would unfold them. So that's Lindquist's area of special interest, protein folding.
It's a wonderfully useful tool in biology. It actually comes from a jellyfish, but it has this lovely property of glowing green when you shine blue light on it. And we use it as a tool, as you'll see a little bit later in the talk. But you can imagine that it's kind of complicated for these proteins to fold up into these shapes. But the problem becomes much, much greater inside the crazy environment of a living cell, where the concentration of other proteins is about 300 milligrams per mil. Now that's about the concentration of protein that crystallographers have in packed crystals when they are actually exploring the protein structure. But of course it's not like a packed crystal. The cell is very, very dynamic and proteins are moving around and colliding into each other all the time. So when you think about proteins going about their business inside of a living cell, it's not like Fred Astaire and Ginger Rogers. more like the characters in a Marx Brothers movie, where chaos is poised on the precipice of disaster. So that's what
I love that bit. So yes, again, the um, what's going on inside the cell is not some orderly, beautiful sequence of movements reminiscent of Fred Astaire doing ballroom or whatever that is. Um, but life processes use as much disorder as possible in the elemental motional processes that are added up to what we think of as directional movement. They're the actual things that are driving them, the events that are creating them, are as disordered as possible. Life is always operating as close to that maximally disordered state as can be accommodated. And it's for energetic reasons. It's very inefficient to organize things against the general background tendency to disorder. It takes a lot of energy to do that, and so it's used in a very clever way in biological systems. As a result, people don't have an intuition for what happens at the subcellular level. The things, the kinds of movements that are adding up to these creepy amoeba movements they're not familiar to everyday experience. They're not furnished by everyday experience. Um, so there's the classic. So we've looked at amoeboid movement. We've talked a little bit about how paramecia move, and I'm asking again this question: where does the movement come from? And if you say it's from like these specific proteins that are doing something, then it doesn't necessarily answer the question because what's making the proteins move? Proteins themselves are these kinds of origami like things, these pieces of origami that somehow know how to fold themselves into the shape. That's the protein folding problem, which I had some interest in and worked with a little bit some people in that area. Um, but the self motion does that answer the question? Are the proteins moving themselves? It's the thermal motion, and so to really understand that answer, we need to understand the thermal motion, and it's not a it's not a simple thing to necessarily get exactly right. That if you put a drop of food coloring um, over here at time at time zero, that over time (20 seconds or minutes or hours or whatever), eventually it spreads until it fills the whole area evenly. It's diffusion. The process is called diffusion. Diffusion, the movement of a substance by random thermal movements. Movement um, by random or thermal microscopic movements. So when we put that drop of dye over here, it looks like a drop of dye over there, and it's not everywhere because the molecules of the dye have a different quality. They interact with light differently, and they're all together. After, but each drop of dye, if you zoom in on it, many times, zoom, then um, it's say a molecule. It's got an actual structure of some kind. Fake. What am I drawing? It's like a phenyl ring and then some cyclobutyl garbage. Anyway, so this group of atoms that are bonded together into this thing, they don't know that they're starting off close to other ones necessarily. They could be interacting with each other a little bit, but really there's all these water molecules that, if this is like on the edge of the droplet, there's no reason for these water molecules to stay here and to not bump off of it and eventually go there, or for this thing to stay where it is and not to be rotating a little bit that way or that way. And so for more water molecules to get in behind it. And the point's that the random movements that are happening at the thermal level, they add up over time to something very non-random. This is the important thing, is that this, when we put this drop of dye over there, it's organized into a specific location. There's something very non-random about the location of the whole droplet of dye. And um, but the movement of all the dye molecules in the water, it is completely random. And so it's only a matter of time before it spreads out and eventually fills all the space. And when it fills all the space, then the information is gone. You don't know where it started necessarily. Well, actually, you necessarily don't know where it started from after you've waited a long time, because the mixing's completely even, and all the parts of it look the same, and all the movements are still random. So there's seemingly no way to get back to. You could play the movie backwards and demonstrate that if time did move backwards, you'd get back where you started, but there's no way to actually ask all those dye molecules to reverse their courses and do that thing. So that's what causes a neurotransmitter to move across a synapse. That's what causes a myosin head to move a little bit forward over the surface of an actin filament. That's actually what's causing the movement, is this random thermal motion. So the same processes that drive diffusion are driving all these biological events at the bottom. And then biological structures and biological systems harness these diffusion type movements, these random movements, and direct them in specific ways. Um, so I was going to show the flycasting pilot. This is something. Um, wow, why did the color do that? Oh yeah, because I've got all these video effects on. So yeah, I'll turn those off. It still seems zoomed in. I thought I turned the crop off. Well, this is entertaining. Anyway, I think I might need to restart the whole thing to get it to uncrop. It seems to have lost track of what it's doing. Yeah, there we go. So this video was made in the lab of Brian Sykes, down there in the Watermark, at the end of my PhD work, mostly with the labor of Jocelyn Dombroski, um, who was also working in the lab at the time. And the whole thing's a model. It's not showing actual simulated data, and the model is intended to put together some observations that the Sykes lab had contributed around this time. Um, Notably, my author Tareen Blumenschein had studied this troponin core complex, the red, blue, and green parts, not all the blue part, but the blue part that's chopsticked with the yellow chopsticks. Um, Studied that soluble chunk. That's actually a good view of it. I'll just pause it there. So Tareen had taken the starting here on the blue part and the whole yellow part and the red part, and studied that with solution state, nuclear magnetic resonance spectroscopy, uh, and not just studying the protein, but studying the protein that was per-deuterated, that had been produced in an environment where most of the hydrogen atoms were replaced by deuterium, which is an isotope of hydrogen, but is nonetheless not exactly chemically equivalent. And so, long story short, it is a project and a half to put together the sample. Very, very ambitious project done in collaboration with, I think, the Frederick group. And uh, the observations showed that for this yellow protein, troponin I, the end of it, the C terminus, was disordered. And so this video shows an integration of all these observations that I published with Brian and Tareen. And so that's this. Yeah, this article, an interplay between protein disorder and structure confers the calcium regulation of striated muscle. So, in 2006, I helped get some people over the resistance to say that the alternation between disorder and order in the different parts of this protein switch, the protein switch comprised of the red, blue, and green, red, blue, and yellow proteins, that this protein switch actually alternated between highly organized and disordered states, with the movement being, in most cases, functionally relevant. At least all the movement that's depicted in this cartoon here is supposed to be functionally relevant. And again, this is based on data that was integrated mostly from a single set of studies, but then combining them with information about the rest of the muscle filament. And so, just to name all the parts, I'm not going to explain all the things, not even close. But the gray thing is actin, and the black thing is tropomyosin, which is helping couple the troponin, the red, yellow, and blue thing, that three-part protein. It's coupling that to the actin, and the tropomyosin is doing probably other things related to the regulation of the muscle that I'm not going to get into. But the point is that when the yellow thing is stuck to the gray thing, when the troponin I is stuck to the actin, then a protein you can't see here, myosin, cannot interact with the actin properly to generate force. So if my arm is the actin, the gray thing, then myosin, my fist, it generates force by locking onto the actin filament, and then an energy producing an energy providing molecule, ATP, attaches to it, allows it to come off of the surface and start diffusing. This is observations from Dave Thomas's group mostly. And then it spontaneously will attach again, and when the ATP is then hydrolyzed, it will lock on. And so the low energy state and the actual process of latching and moving forward happens like based on the actual structures of the things. And the moving forward part is again random thermal motion that is allowed to happen because it gets released from its previous latching point by the binding of ATP and probably some other things. But most in terms of the causal chain, most directly the ATP. So although I'm changing subject from regulation to contraction itself, that's why dead people get stiff. That's why stiffs are stiff, is that the resting state of striated muscle is in the contracted state. It's holding things in the contracted state is actually the resting state. And for things to relax again and to well, not for things to relax again. Sorry, it's not the contracted state. The resting state is the latched state. The myosin's locked onto the actin, and then for it to be able to move, it requires the presence of ATP, which is only around in significant amounts when the organism's alive. So yeah, the energy source that's being updated will refresh, replenish continuously as the cell lives. So what we're seeing here is the starting process of where the movement happens in the example of voluntary movement. If you're doing your bell hand thing, then your neuromuscular system is recruiting voluntary movement. And the neurochemical signal from the neurons is going into the muscle cells. It's causing a release of calcium from a store inside the muscle cells called the sarcoplasmic reticulum. And so that's that white sphere that's drifting in here. The calcium ion sticks to the one end of troponin C and causes it to open up. So a movement of the calcium ion, which is random, sticks to this simple switch-like thing, which then opens up. And then when troponin C is opened up, it's able to then bind to this part of troponin I and remove it from well, first it binds to this part of troponin I, and then subsequent changes allow the rest of the troponin I to come off of the actin surface. And then once that happens, there's movement in tropomyosin, which we're not showing in this video, that allows the myosin head to get back at that actin surface. And so it's being blocked when there's no calcium and the troponin I is on the muscle. When there is calcium, then the myosin is not being blocked and the contraction can occur. So I figured out almost none of that. I didn't even figure out the fact that the movement is occurring in these various places was cobbled together from multiple papers in my 2006 paper. But the basic calcium regulation of muscle contraction was figured out by Ibashi in, I think, around 1950, a Japanese biochemist. So, oh no, that'd be more like 1970, I think, Ibashi. Because yeah, the sliding filament model is more like 1950, I think. So actually, I think I'm wrong on the sliding filament model too. Let's take a look at that because I have found this reference here. Here's a sliding filament model review from 72. Oh, so mid 50s. Okay, yeah. So it was 50s the sliding filament model, and and the regulation of striated muscle contraction by troponin is not in this review. But that myosin is the motor protein has been known for some time, and how that might work has been an ongoing area of research that is reviewed in articles like this. this is just a part of the whole picture, but um, this is the thing that's actually capable of binding directly to actin. Um, over here, I guess you can see the various annotations of the parts of this machine, and it's yeah, maybe not that intuitive how this thing functions. So what you can do is you can begin to capture images of the functional machinery in different contexts. And so here we're seeing a little bit of the actual where the force comes from, where the force comes from. And you can see that if you compare these two structures, that there's a large change in the orientation of the purple, yellow, blue bit with respect to the red and green bit. And so the I think the blue and white stuff over here is actin, and then these are various parts of the myosin, and they're showing how the movement the movement in the rest of the myosin is latching in. It's latching in after the binding site on actin's been found. So the myosin finds a contact site on the actin, and then movement causes the subsequent movement is built into the actual mechanics of the protein itself. And again, all those forces are being transmitted through the protein. Well, that's just normal mechanics from that point. Just like the opening of troponin was just mechanics. But again, the initiation of these events is still, you're going to be able to show that it's random thermal motion is what's actually the underlying driving process for all this. That would, for example, convert the A image into the B image. The article purports to be about the sliding filament model, but it's actually much more about force generation by myosin, I realize. So I'm not going to review the sliding filament model. It's in all the physiology textbooks. You can find that probably fairly easily. But the types of movement that are being cartooned here, um, they have their own kind of intentionality that's, well, like the calcium ion manages to go in the right spot. And it's just hard to appreciate that it doesn't know where to go, and that the calcium's just moving randomly, and that there's enough calcium released into this compartment that its ability to find this binding site is guaranteed. It's as guaranteed as when you put those droplets of dye in one area, one end of the pool or whatever, that it's guaranteed that over enough time they'll find every spot in the pool. The quality of the movement we're looking at here, it isn't supposed to be the true movement that's happening at the molecular scale. But it was characterizing enough of the essential movement that we could get our point across in terms of which parts are rigid and relatively ordered with respect to each other, and which parts look like they're moving independently of the rest of it, waving around independently compared to the rest of the structure. So putting this together in a coherent way, yeah, that was a contribution here. Blah, blah, blah. What? So I think that Susan Lindquist video says it well, that or illustrates well with that joke, that what's happening at the cellular level, it's not easy to picture as being ordered, organized processes, and understanding that the bustle and chaotic motion is beginning to take over and be the dominant part of the picture at the molecular scale. It's helpful. It's helpful to internalize that idea. There's I'm looking for a demonstration of Coette flow. This looks like a nice apparatus. I haven't watched this video before. Hopefully it's not porn. All right.
Hey, it's me, Destin. This is the second channel. We thought it'd be fun to do a Taylor Coette flow shot that was nothing but one tripod setup so that you could see the experiment just go down, and then you could see it time-lapse and sped up and stuff like that. It's a really neat experiment, and it's a really fun thing to watch. So let's do that.
So the apparatus is like a cylinder inside a cylinder, so that there's a little like a donut-shaped profile of liquid around the outside of that white cylinder. And then he's carefully pipetting into it some colored dye, and the liquid is viscous. It isn't water that he's pipetting into. He's pipetting a liquid. They're probably both viscous, and he's pipetting something that's viscous into another thing that's viscous, like cornstarch or something like that, so that it's not immediately going to mix like that droplet of dye in the swimming pool. But it is. It's still automatically mixing from thermal movement. And so to make it beautiful and more compelling, he's adding colors. And it's the same idea. When he introduces this green dye, it's going to stay put. It's going to stay where he's putting it to a good approximation, because it's a very gooey liquid that's being used for this demonstration. And the gooiness is very important. It slows down the rate of randomization. It makes it so that the scrambling from random thermal movement is less quick to occur. He's very precisely laying out a shape, or he's trying to very precisely put the glob in there so it's well defined and has an interesting shape to it. At least I think that's the intention. There might be some technique here I'm unfamiliar with. All right, this is an 11-minute video, so I'm going to jump a little bit forward. I think I saw that there's a third color he's going to introduce. Yeah, there's a third color he's introducing. And so you can see some of the hints that it's a very viscous fluid. As he's moving stuff around here, these other things are swaying in response. But now we've got the 3 bands of liquid.
Here we go.
So when he turns that handle, the inside thing is rotating, but not the outside thing. And so it's causing everything to smear and mix, as you can see. Everything's being smeared and mixed. You can tell that they were separated once after one turn. I like how amused he is.
We're
Yeah.
taking thumbnails if you wonder what we're doing.
Oh no. Apologize. Spare you that nonsense. Spare you this nonsense. All right. Back to our scheduled programming. So it's looking pretty mixed, pretty scrambled. It looks like he's reversing it now. Yeah, he's now reversed. He reversed his direction.
There's no way this is going to come back. I put all three colors right on the edge. There's no way, dude. I basically mixed them up.
They did look pretty scrambled. They did look pretty mixed up. But the point is that this apparatus is letting him non-randomly change the positions of things, and the mixing is coming from the actual random initial phases of everything. That all the dye molecules had initial velocities that were all in randomly different directions. That adds up over the small differences. Add up over these large shifts in displacement to cause mixing.
Dude, this freaking works, man.
And so you can
all against, like straight against the pipe.
So you can unmix them with the Coette apparatus.
Shut up.
By turning the crank the exact number of times backwards, that was the same number of times that he went forwards. He can get back the original spots with very little blurring. The blurring will still occur. It won't be. It won't be exactly perfect, but it's pretty compelling that you can
Like they were all in the same plane around the cylinder.
Mixed and unmixed.
What? It's not all the same type of corn syrup. It's different types of corn syrup too. How did that work, dude?
Do 12 turns right now.
12 turns.
The other way.
The other way. 12 turns.
12 turns.
1. 2. 3. 4.
Hear the birds in the background. It's fun.
5. This is fast too. 11. There's no way, dude.
It looks very thoroughly mixed.
12. This is not clickbait. This is actually okay. Here we go. 12 turns back.
He's so ginger about it.
1.
Hurry up, dude.
It's going to be much wispier at the top, but it's going to work.
7. 8. I can see it coming back. 9. 10. 11. No. It's not happening. No.
It's there.
12.
Oh my gosh.
No, it's not.
It's definitely more wispy, but yes, it's there.
How the heck, man?
So this is how, is that in a Coette apparatus, you can separate out the random from the non-random aspects of mixing. And the non-random thing, you can demonstrate that it's non-random because you can reverse something that's non-random. Reversibility is very important. So if you put dye in a swimming pool and just let the dye diffuse across the swimming pool, you know time's moving forward and things make sense. If you put dye in the bathtub, if you walk up to a bathtub and it's full of food coloring, and then the food coloring concentrates itself in one corner of the bathtub and then jumps out of the bathtub into your hand, then you know something's wrong. Time is moving backwards, something's not right. So life processes are continuously using this ability to borrow randomness when it's helpful to get things done. Life processes have all kinds of intelligences, all kinds of wisdom reflecting what to do about things being disorganized. In Laban Bartinieff analysis, at least what I remember of it from Margarita Kozhevnikova, um, um, one can think about the quality of a movement. You don't just express the direction of things, but also the intensity, the amount of force, things like that. How one actually expresses with movement, it matters. It's significant. And so the thing I want to kind of rest on here, which is not exactly a point that I've made very well, is that the kind of movement that we're looking at the cellular level, at the subcellular level, the kind of movement that that causes the larger scale movements in a direct way, it's not Fred Astaire style smooth movement. It's not it's not it's not elegant voluntary movement that's being recapitulated in a fractal way. So people like fractals and thinking that things are reflected on many scales. And in biology, there are phenomena that are multi-scale that way and that appear to be consistent with that kind of organization pattern. But movement quality itself, it doesn't fit that. That what I'm doing here is not like what my cells are doing down there. That the cellular movements, I can move in a way that imitates them. All of the cellular movements are consistent with me, but the large scale movements that my organism can do, these are not things that are analogous or recapitulated on the scale of the cells. So there's in body mind centering, there's this popular image of cells contracting and expanding with the respiratory movements. And just because something feels nice doesn't mean that it's educationally constructive. And I think thinking that your cells are fractally doing something that's analogous to what you're doing in a breathing movement is, yeah, counter-educational. So the good news about our cells' respiratory movements is that they happen spontaneously, driven through thermal movement, and no voluntary movements required to get the cells to do their things. if you put a maybe this video is good. Yeah, these aren't great videos, but never mind. You take a single cardiomyocyte, a single heart cell, heart muscle cell, you put it on a petri dish, and it just beats. It's the physiology of a cardiomyocyte is such that it's going to undergo striated muscle contractions according to its own signal, its own rhythmic pulse. And it's only when you grow enough of them that they start touching each other on the plate that their rhythm starts synchronizing. But that's all cardiomyocytes need to do what they do, is that they grow until they're touching each other, and then they all beat together. They just feel the presence of each other, and they move in synchrony. That fetal heartbeat you hear about in abortion debates, that's the same thing as the existence of cardiomyocytes that are undergoing these contractile cycles. That again, they'll do in a test tube. They'll do outside of an organism, outside of a multicellular organism. So those are cells that have a pulsating movement to themselves. And their movements are adding up in a kind of fractal way to the larger scale movements. So there's your example. There are ways in which things will scale that way. But that is the exception and not the rule. That is a very specialized tissue doing something almost miraculous. That's what I had to say about that.